So I'm really honored to follow all these amazing talks by these eminent members of the society. But I was just telling David, well, first I'm going to say, do you guys think you can handle 2 Canadians in a row? And David forgot to mention Canadians who have been, I think, played an important role in the society, including a founding member of the society who was a co-author on that Committee 17 report. Can anybody tell me who? Who? No, this is way before that. Jack Van Borstel. And of course, many other Canadians. As our former lab director, now retired, David Blakey used to say, we're small, but we punch above our weight class. So, and Errol was saying that we like to be informal. So I say Errol, not Dr. Zeiger. So I'm going to thank Mimi and David for giving me the honor to present to the entire EMGS community on the auspicious occasion of the 50th anniversary. And I'm going to talk to you this morning about the shifting paradigm in genetic toxicity assessment. So what I mean by that is the shift from qualitative hazard ID to quantitative interpretation of dose-response data in support of risk assessment and regulatory decision making. So to answer this question, how much is too much? So I'm going to start with a quote. Sola dosis facit venenum. You can tell me what that means. I orchestrated that, but they were late in responding. Dose makes the poison. So this is the 50th anniversary. That's a 500-year-old quote. Here's the more full quote, translated from the original German. All things are poison. The dosage alone makes it so a thing is not a poison. Paracelsus. So that's 500 years old. And we heard, I think, from Errol that gene tox is a subdiscipline of toxicology. All of toxicology has adopted this for hundreds of years. But in gene tox, we do hazard ID. So we do qualitative screen and bin. And here's what I mean. So this is the traditional paradigm. We do a test. We look at the test results. We bin it into no evidence of genotoxicity, the green bin, the good bin, or evidence of genotoxic activity, with no dose-response analysis and no attempt to interpret the dose-response data to determine what the risk assessors would call a health-based guidance value, so a human exposure limit below which the risk of adverse effects in humans would be negligible. We just don't do that. Well, we don't do that with moose. No, sorry. So this paradigm, the old paradigm of qualitative screen and bin, I'm going to argue, is predicated on 3 assumptions that are false. And we heard a bit about this from David and from Errol, I think, that mutagens are rare. They're not rare. And that exposures can be entirely avoided. They cannot. How many people had a cup of coffee? 5 nanograms of BAP, benzopyrene, per cup of coffee. So there you go. You cannot avoid it. Secondly, that genetic toxicity dose-response data do not provide evidence of thresholds. That's not true. So when we think about, is there a safe level of exposure? Well, I don't want to use the word safe, but there will be an exposure level below which the likelihood of an adverse effect is negligible. Third, that mutagenicity is not or should not be regarded as a bona fide toxicological endpoint for regulatory decision making. So it should only be used for identification of potential carcinogens. I would say that that's also not true. So mutagens are not rare. This is the first one, number 1. Think of mutagens as natural origin. That goes back to, what did you say, David? Charlotte Auerbach, to the 40s. So this is a funny slide. Here's a Thanksgiving Day menu. This is what you would eat at Thanksgiving. In Canada, it's only in 3 weeks. Turkey dinner, side dishes, salad, bread, pumpkin pie. Here's a list of the mutagens you'll be exposed to if you participate in this Thanksgiving Day dinner. Many of them are potent, and I'll highlight in later slides benzopyrene, the product of incomplete combustion. It's in the context of this dinner. It's in the bread, it's in the pie, it's in the coffee, it's in the tea. You cannot avoid being exposed to it. So very quickly, the second one, what about thresholds? For those of you who've looked in the threshold literature, you've never seen anything so complicated, confusing, and convoluted as the threshold literature. All these terms are used. I prefer just threshold, or maybe biologically meaningful threshold. I would argue, and I'll show you here, this is a slide from Alexander Long's PhD thesis, a very fat PhD thesis. This was her experiment that we like to call the mega BAP experiment. These are transgenic rodent, transgenic mutant mouse exposed to 10 doses plus control of benzopyrene in a 28-day repeat dose oral experiment. And here you can see LAC-Z mutant frequency along the y-axis against dose, and this is 7 tissues. That's what those 7 lines are. So what you can see here, there's a flat portion of the dose response. So I would argue that that's the point at which the chemist would call, or the pharmacologist, zero-order kinetics. There's no change in response with increasing dose until you reach this inflection point, which some people would call the threshold. Beyond that, you see an increase in response with increasing dose. So we see that. So I would argue that there's evidence of a threshold, but some people are really picky. They want to have mechanistic understanding to justify that word, threshold. So you see that. You see that. Here's an experiment that comes from Swansea University, and here you see compensatory responses, in this case base excision repair, that actually define the threshold. So you can see you've got micronucleated frequency, micronucleated cell frequency in AHH1 cells exposed to ethylmethane sulfonate. You see a flat portion there in the control plot. There's no response with increasing dose until you reach that inflection point. You could say that's the threshold, and then there's an increase. Now what happens if you knock down MPG, methylpyridine glycosylase? The threshold is pretty much gone. It shifts so far over to the left, you cannot even see it. So you've shifted the threshold by blocking the compensatory response. So the compensatory response is defining the threshold. We now have biological understanding of the mechanism underlying this threshold phenomenon. And we can quantify that. That's what it's all about, quantification. You can calculate what we call point of departure metrics, like no gel, no observed genotoxic effect level, or benchmark dose. That's the dose, in this case, that results in a set fractional increase above background for a given, the dose that causes a set fractional increase above background. And you can see here for the control, it's 1.3. So that's the point at which, that's the point of departure, or threshold, if you will. When you knock down MPG, it goes down to about a fifth or a third of that. So now we've shifted the threshold way over to the left, and we have mechanistic understanding for that threshold phenomenon. So I would argue that we could say, I don't like to use the word true, or biologically meaningful. So what about the number 3? What about recognition of genotoxicity as a bona fide toxicological endpoint? I'm proud to say that Health Canada was quite prescient, and in the 80s and 90s actually declared that this should be the case. We should regard mutation as a bona fide toxicological endpoint for regulatory decision making. And actually the mutagenicity guidelines were published in EMM in '92, and George Douglas, who's here today, was involved in that. And here's a short quote, I won't read the whole thing, but this is a quote from the publication. Genotoxicity is a bona fide toxicological endpoint, and notwithstanding the correlations that have been observed between mutation and cancer, it's likely that an agent that induces mutations in somatic cells of test animals will possess the potential to cause adverse health effects other than cancer. So as we heard from Carol, we know that there are diseases related to mutations. She talked about heritable genetic diseases, and there's a human gene mutation database with over 250,000 entries. But what about somatic cell damage and diseases other than cancer? It's a bit hard to read this table here, but it's from Yusufian and Piretz in their Nature Review Genetic paper. And they list a series of diseases, categories of disease, and the individual diseases that are related to somatic mosaicism. So this is damage to somatic cells that result in a distinct population of cells so that the organ and the organism is mosaic, and there's some sort of phenotypic change that results in sufficient dysfunction to confer disease. So we have diseases other than cancer that are conferred by somatic damage. So that leads us to a new paradigm. So I hope I've convinced you that those 3 assumptions are false. So we need a new paradigm where we can move forward with quantitative dose-response analysis for risk assessment and regulatory decision-making. So here's the conceptual framework. It's very straightforward. I borrowed this from Véronique Thibault at Sanofi. And here's the conceptual framework for the quantitative analyses. It's quite straightforward. We have genotoxic activity is the response. We have dose. We have a point of departure like the benchmark dose. This is the dose that causes a fractional increase in response. We can invoke the use of uncertainty factors. This gets a little complicated. And then we arrive at what's called a health-based guidance value. So that's a human exposure limit below which the risk of an effect in humans is deemed to be negligible. So that's what we can use for regulatory action. So risk assessment and regulatory decision making. Dose makes the poison. And we're assuming, as I showed, I hope I convinced you, that at low doses we have these cellular protection mechanisms that are efficient and not saturated, and there's low doses where there's no difference between the response and the background. It's negligible. So, you know, we use this expression in English for those of you who are not native English speakers, where the rubber hits the road. So where the rubber hits the road is putting this into practice. So I'm just going to borrow from a paradigm that ICH has put forward for things like impurities in therapeutic products, and they calculate what's called the permitted daily exposure, the PDE, when there's sufficient evidence for a practical threshold. And this is the equation they use. So here they've got no L or low L, in this case no gel or low gel. You use body weight to calculate the per individual dose that's this point of departure, and you divide it by the product of a number of uncertainty factors. So I would argue that this should be replaced by BMD, and you have BMD for the most relevant animal study or endpoint, and you divide it by the product of a series of uncertainty factors. You have to come to the session on quantitative analyses on Sunday, and I'm giving a talk with Stefan Fuhler where we talk more about these uncertainty factors, very controversial. So you need to do species extrapolation, inter-individual variability, uncertainty correction, study duration if the study is not sufficiently long, correct for or adjust for effect severity, and then database insufficiency, which doesn't apply if you're using benchmark dose. So we calculate a permitted daily exposure as the ratio between the point of departure and the product of a series of uncertainty factors. So let me show you how this would work in practice. I'm going to get back to Ali's benzopyrene example. So here we have the same dose response that I showed before. So it's laxi mutant frequency in mutamouse against dose in milligram per kilogram body weight. And now I'm going to show you the tissues though. So here's small intestine is the most sensitive tissue, the lowest point of departure, the lowest benchmark dose. Kidney is the least sensitive tissue. So we should argue that we could extrapolate based on the small intestine results to be conservative and determine a human exposure limit based on that sensitive tissue. I think it's safe to say that we have evidence of a threshold and large differences in potency across tissues. So I'm going to walk you through how this calculation might look. So here's the lowest BMD 50, so the dose that results in a 50% increase over background. And very importantly, you have the confidence limit. So that's the lower and upper confidence limit, 0.32 to 0.54. So I had to show some data here because it's about quantitative analyses. So what you can do is you can use allometric scaling. This is the interspecies adjustment factor to calculate the human equivalent dose given a body size of 60 kilos. Might be a bit low these days, but that's what we use. So you get 1.6 to 2.6 milligram per person per day. Now we need to adjust it using the product of these uncertainty factors. A lot of debate about what they should be, but for illustrative purposes here I'm saying 10, 10, 10. So it's 1,000. It's very simple to do the calculation. You take your 1.6 to 2.6 mgs per person, and you just go down to micrograms per person. So that's the tolerable daily intake. So I'm using a different term here. It's always very confusing in the literature with respect to these point of departure metrics or health-based guidance values. The pharma guys use permitted daily exposure. The industrial chemical folks use tolerable daily intake. So the tolerable daily intake is 1.6 to 2.6 microgram per person per day. Now in your cup of coffee, you have 5 nanograms. So you need 300 cups of coffee to get up to the TDI. How many people here drink 3 cups of coffee a day? 1% of the TDI. For every 3 cups, 1%, 2%, 5% would be whatever, 12 cups of coffee. I probably drink that much. So what you want to do from a risk assessment point of view is you need to compare it to exposure. So how much benzopyrene are people exposed to? So if you look in the US literature, the dietary intake is 0.04 to 2.8 microgram per person per day. So how does that compare with the tolerable daily intake? Well, look at it. The average person is way below. But what you can see there is look at the 2.8. So those people who are at 2.8, they're even above the upper confidence limit of the TDI. So who are those people? I don't know. Texas barbecue, 3 meals a day. But there are some people who are way above the TDI, but most people are not. It's a very skewed distribution, but most people are not. So there's my example. I've turfed the 3 assumptions. I've given you an example. What we need to ask now is, are we there yet for routine use of genetic toxicity BMDs for human health risk assessment? I wish I could say that we are, but we're very strict in terms of our requirements for putting this into practice, and there are what I would call tough nuts to crack. So there's 2 tough nuts to crack. One is what we call definition of an endpoint specific critical effect size. So in the example I showed you, we're looking at a 50% increase over background. So the dose that causes a 50% increase, called the BMD 50. Is 50% right? That's the critical effect size. We may need to evaluate that. Secondly, we need to scrutinize those uncertainty factors. I showed you 10, 10, 10, and Stefan and I are going to talk about what we might consider. So empirical analyses of literature data, for example, to get a handle on what those could or should be. So I'm just going to wrap up. I'm kind of out of time. We've come a long way in the quantitative gene tox dose response analysis community. We didn't have anything. We didn't even know how to analyze the data. Well, we knew there were paradigms from other tox endpoints, but we started from scratch and we've done a lot of work. Where the rubber hits the road is risk assessment and risk management. We need case studies, like I did for benzopyramine, to demonstrate the utility of quantitative analysis, and I'm saying of in vivo mutagenicity data. In vitro is a whole new set of problems to do in vitro. So there's really an acute need to determine the adjustment factors. I want to show this last slide. Oops. We need to scrutinize literature data, I would argue, to try and come to grips with these adjustment factors. And I'm going to end with 2 additional quotes. This one from someone in the room here, Bob Heflick, from a paper that's now in press in EMM. Implementing quantitative methods for human health risk assessment will make better use of mutation data, will be of value in mitigating the negative health outcomes of environmental exposures. And that's a commentary that's coming out in the 50th anniversary issue of EMM. And I'm a co-author. I'm proud to say that several people in this room are also co-authors. I'd like to end with a quote from Lord Kelvin, William Thomson. When you can measure what you're speaking about and express it in numbers, you know something about it. When you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. It may be the beginning of knowledge, but you've scarcely in your thoughts advanced the stage of science. Thank you.